learning outcomes. After studying this module, you will be able to know about hair as evidence, about use of microscopy for hair examination. Introduction. Follicles are present in the skin or dermis from which protein filaments grow. These protein filaments is known as hair. Human body is covered with skin and hair follicles within the skin. These follicles produce both thick and fine hair depending upon the location of hair on the body. Hair is composed of protein called keratin and is an important biomaterial. Hair is one of the common and important physical evidence encountered in a crime scene. Individualization of human hair, that is, whether it is from head or body. Forceful removal of hair may have blood or skin with the root. In such cases, DNA typing can be done. There have been advancement in the DNA typing technology. Earlier, only hair with root attached could be used for typing, but now with advanced technology, hair without root can also be used if there are enough number of hair strands. In absence of root, blood or any material through which typing can be done, that is the appearance or morphology, that is the structure and color of the hair, becomes the most important characteristic feature in forensic science. The proper collection and examination of hair can provide strong corroborative evidence. Differentiation between cut, forcibly removed or fallen hair can be made easily. Hair is an appendage of skin that grows from the hair follicle. It is root, bulb or a knob, a shaft and a tip. The portion of the hair that lies in the follicle is known as root. Root is surrounded by the loose connective tissues known as the root sheath. It lies in dermis. The shaft grows and projects out of the skin. The distal end of the shaft is known as tip. Vacuoles or air bubbles are present in hair. It consists of three zones: the cuticle, cortex and medulla. Examination of these three zones of hair is done for the identification and the comparison purposes of hair. Simple microscope can be used to observe hair and its characteristics. Now about the forensic importance of hair. Mutual transfer of hair due to contact is a classic example of Lacat's principle of mutual exchange of the traces. Hair, if properly searched for, are found in large number of cases. It is one of the earliest types of physical evidence which are considered, collected, evaluated and utilized in the forensic caseworks. Individualization of hair with regard to its source has been eluding the scientist up till recently. The identification of hair is not conclusive at the present stage of development except through the DNA typing, if root hair are present, or through mitochondrial protein analysis. In extremely rare cases, the presence of some individual dye, contamination, some extraordinary defect or disease in the hair, however, may permit the individualization of the source. On the other hand, it is possible to exclude some of the persons as the possible sources of the questioned hair. This exclusion, the possible corroboration and its identification now with the new techniques, makes the hair evidence one of the important pieces of physical evidence. The evidence establishes the possible presence of the culprit at the scene of occurrence of crime. It indicates the contact of transfer of hairs has taken place. It indicates the routes of ingress or egress if some hair are retained at the point of entrance or the exit. It helps to identify the vehicle used in the commission of crime. It may also be useful to establish the ownership of the articles left at the crime scene. The hair evidence is often not easily seen, hence it goes unnoticed and it does not bother the culprit. Yet, if they carry blood or blood cells, through DNA typing, they permit the definite identification of the culprit. Hair evaluation have always been utilized to determine the nature, the quantity and the period over which the poison had been administered in slow poisoning cases. In case of Napoleon, the nature and quantity was determined after about 140 years. The hair evidence as a corroborative evidence is available in most of the serious cases, especially in offences against person. Every human being sheds hair, normally about 10 hairs a day. The falling rate if person is under emotional stress, such as in rape or murder attempts, increases. The hair are therefore likely to fall at the scene. They can form important corroborative link if collected properly. The hairs are ordinarily available in murders, assaults, road accidents, rapes, abortions, bestiality, housebreaking, maiming, poaching, worrying and thefts of fur and animals. The identification of hair is not an easy task. Extensive experience is required in this field as there is variation in the hairs of the same person at some as well as the different time. Further, there are similarities in the hair of the same person. However, recent advances, especially in DNA typing of hair root cells and the mitochondrial proteins, hold promise for definite acceptable conditions whenever body cells are available. Next, we will learn about the microscopic examination of hair. Light microscopy is used for examination of human hairs in forensic laboratory. The two steps are followed in this examination, that is the identification of the questioned hair and the comparison of the questioned and the known hair. The microscopic examination is conducted so as to analyze whether the two or more individuals came in contact or a person came in contact with an object. The value of hair evidence is related to the variability of the hair characteristics between the individuals and the populations, which can be visualized through the use of the comparison microscope. Various factors affect the reliability of the hair association, which includes experience, training, adequacy of equipment and suitability of the known hair standards. Although the hair evidence is a valuable tool in human identification, it is difficult to establish a statistical probability for a particular association due to, in part, to the lack of the reliable quantitative assessments of the microscopic characteristic present in the hairs. Two compound light microscopes are connected by an optical bridge in the comparison microscope, which helps in the simultaneous weighing of the questioned and the known hairs. A glass microscopic slide with known or the reference hair sample is placed on the stage of the other microscope. This helps in comparison of the microscopic characteristics of the known and the questioned hair samples. The range of magnification commonly used is approximately 40x to 400x. Various steps are involved in examination of hairs. First examination is to check whether the hair belongs to the human or animal, and if it belongs to animal, then which animal it is possible to identify the animal. Although certain hairs can be attributed to species, it is not possible to identify hairs to a specific animal to the exclusion of some other similar animals. Next, we have the difference between the human and the animal hair. There are some of the characteristics that are observed when microscopic examination of hair is done so as to check whether the hair belongs to an animal or an human. In this table, we have shown the various characteristic features and the comparison between the human hair and the animal hair. In the characteristics part, we have the cuticle, medulla, cortex, medullary index and the pigment. Next comes the various types of microscopes used for the hair examination. Presence of blood or debris on the hair samples may be significant. In case the material adhered is of evidentiary value, then it should be removed and preserved. If it does not have an evidentiary value, then the hair can be washed and cleaned before mounting it. The presence of a small amount of debris or blood on hair may not interfere with the microscopic examination. Cleaned and washed hair should be dried before mounting it. Those hair samples which show thermal or mechanical damage may be more brittle and should be handled carefully. The various types of microscopes that are used for the examination of hair are: first is the stereoscopic microscope. For initial examination of mounted or unmounted hairs, the stereo microscopes or the stereoscopic microscopes are used with a magnification range of up to 100x. Then next comes the transmitted light microscope. For identification and examination of hair of high quality, the transmitted light microscope is required. The objectives and the eyepieces should permit the observations in the range of approximately 40x to 400x. The examiner's ability to observe certain features is examined by using a polarized light microscope. Next comes the comparison microscope. While the comparing microscopic characteristics here, we need a high quality transmitted light microscope. The high quality objectives are very important. The objectives and the eyepieces selected should permit observations in the range of approximately 40x to 400x. A high intensity tungsten light source suitable for the photomicrography and equipped with the daylight correction filter, which is present for providing adequate lighting. Both sides of the comparison microscope should be balanced for light intensity and color. A comparison microscope may be equipped with one of the several types of stages. Now we will study about the observations of the microscopic examination of hair. Preliminary microscopic examination is carried out without cleaning or mounting the hairs. It reveals their color, the contamination and the character, whether they are curly, wavy, soft or coarse, whether the ends are frayed or have tips, or whether the roots are present or not. It is possible to say whether the hair have been pulled out, cut or fallen. The pulled out hairs have live bulbs and signs of stretching near the bulb, while the fallen hairs have the roots that are frowned and shrunken. The detailed microscopic examination of hairs is done after cleaning them in alcohol ether mixture or the acetone. They are then mounted in Canada balsam on a microscopic slide and examined with a magnification of about 400x. The examination reveals the actual color of the hairs. The contamination, which may be masking the real color of the hair, is removed in the cleaning process. Then whether the hairs have been dyed or bleached. Natural color of the hair is found near the root end. The portion of the hair may be long or short depending upon the period class between the dyeing or bleaching and the collection, that is from the head or the fallen hair of the hairs from the examination. the part of the body from which the hairs have come. The head hairs are like rods, whereas the hairs from eyebrows or eyelashes have pointed tips. The beard hair may be somewhat flattened. Then comes the morphology of the hair. It indicates the presence or absence of and the nature of the medulla and the pigmentation pattern. The medullary index, the ratio of the diameters of the medulla and the hair measured at the thickest point, is known as the medullary index. Its determination establishes the species of origin. The medullary index of the human hair is commonly less than 0.3, and in animals it is generally more than 0.5. The medullary index varies somewhat in male and female hairs and in the hairs from different parts of the body as well. Ordinarily, the medullary index is greater in women when hair from the corresponding parts of the bodies are compared. The male beard hairs have greater medullary index than hairs from the other parts. Medulla may be continuous, fragmentary, or it may be absent in human beings. It may also be masked sometimes by the hair pigment. It is made visible by bleaching the hair. Next is the pigmentation distribution or the pigment distribution in hairs. It is helpful in the study of species of origin and in the identification of hairs of an individual. It is often an important characteristic and permits elimination of certain suspected sources of hair. The next comes the roots and the ends. They reveal whether the hair have been pulled out or not, whether they have been cut, and if so, when they were last cut. When tips are freshly cut, they have sharp cut ends. With the passage of time, the sharp ends gradually become round. The possible nature of weapon with which the hair has been severed is also revealed. With blunt weapons, the cut end is found frayed and irregular. With sharp-edged instruments, the cut ends have sharp edges. Any deformity or disease in the hair. Then the presence of vacuoles. They give characteristic formations in certain species which are useful in their identification. They are known as the air bubbles also. The special treatment, for example burning, hairs may have undergone. Next, we will study about the microtomy. The cross sections of hairs are obtained with the help of an instrument called the microtome. Clean hair is embedded in hard wax, plastics or flesh hardened by the special treatment and sliced. The cross sections of the hairs obtained are placed on a microscopic slide treated with albumin. The embedding material is removed with a suitable solvent and the sections are fixed in Canada balsam. Microscopy reveals the cross-sectional structure of the hair. Microtomy is helpful to determine the pigment distribution, medullary shape and medullary index of the hair. In human hair, the pigment is found concentrated near the periphery of the cortex, close to the cuticles, while in mammals the pigment concentration are near the medulla and the cortex. It also permits proper study of the shape of the cross-sectional area. Now, I'll summarize all that we have studied in this module. Hair is composed of protein called keratin and is an important biomaterial. Hair is one of the most common and important types of physical evidences encountered in a crime scene. In absence of root, blood or any other material through which typing can be done, the appearance or the morphology of that is the structure and color of the hair becomes the most characteristic forensic feature of the hair. The hair evidence as corroborative evidence is available in most of the serious crime scenes, especially in offences against the person. First examination is to check whether the hair belongs to human or animal. If it belongs to the animal, then which animal it belongs to. Although certain hairs can be attributed to the species, it is not possible to identify hairs to a specific animal to the exclusion of other similar animals. Presence of a small amount of debris or blood on hair may not interfere with the microscopic examination. Cleaned and washed hair should be dry before mounting it. Light microscopy is used for the examination of human hairs in forensic laboratories. Microscopic examination is conducted so as to analyze whether two or more individuals came in contact or a person came in contact with an object. Two compound light microscopes are connected by an optical bridge in comparison microscopes, which helps in the simultaneous viewing of the questioned and the known hairs. The range of magnification commonly used is approximately 40x to 400x. The microscopic examination reveals the color, the examination and the character, whether they are curly, wavy, soft or coarse, whether the ends are frayed or have tips, or whether the roots are present or not. The ratio of diameters of the medulla and the hairs measured at the thickest point is known as the medullary index. Vacuoles give characteristic formations in certain species which are useful in their identification. They are also known as the air bubbles. The cross section of the hairs are obtained with the help of an instrument called microtome. Clean hair is embedded in hard wax, plastics or flesh hardened by special instruments and sliced. Microtomy is helpful in determining the pigment distribution, the medullary shape and the medullary index of the hairs. Thank you.